Hey guys, our topic for today is about cholesterol. We will be speaking about the cholesterol synthesis, about the main metabolites, and also about a few of the products that we can get from cholesterol. So what's the function of cholesterol? It serves as a stabilizing component of the cell membrane and blood lipoproteins. Okay, so cholesterol is a precursor of steroid hormones, bile salts, vitamin D, etc. So it is an important compound in the body. The biosynthesis occurs in the cytosol of nucleated cells, normal cells. It is stored in the liver, the intestine, and the adrenal cortex. Now we'll move on to the synthesis, the pathway basically. So we start with acetyl coenzyme A, which we can get from glucose, fatty acids, amino acids, right? And it is carried to the cytosol by citrate. The acetyl coenzyme A is converted to acetyl coenzyme A, which is converted to beta HMG CoA, which is coenzyme A. And that goes to mevalonic acid. Now the mevalonic acid in 3 steps goes to become pyrophosphate mevalonate, then it goes to isopentenyl pyrophosphate, and then dimethyl allyl pyrophosphate, and then finally to squalene and then cholesterol. Now moving on to the enzymes. So acetyl coenzyme A, to the other one is thiolase. Then you have HMG-CoA synthase, which is pretty simple. Then you have HMG-CoA reductase, which is the key control step, by the way. So the rate limiting step, right? All right, then we have I told you 3 steps, and then there's mevalonate kinase, phosphomevalonate kinase, pyrophosphomethylonate decarboxylase, isopentenyl pyrophosphate isomerase, but I don't think it's that important. The important thing is that it goes from mevalonic acid to isoprene to squalene and then cholesterol. So from isoprene to squalene is prenyl transferase, then squalene monooxygenase. Okay. So that would be the synthesis. Now a bit about the cholesterol excretion. So it is excreted into the gut in bile. Here it's converted to neutral sterols, which can be co-prostanol and cholestanol. And this is done by the intestinal bacteria. Okay, now we can speak about a bit about the regulation. So you have induction and repression of synthesis of the LDL receptor, because we know that cholesterol binds to LDL receptors, and also it can be so we have like 2 groups, right? So for regulation we need some hormones or some substances that can increase the production or reduce the synthesis or degradation, for example. So glucagon, if glucagon is elevated, the enzyme is inactive. Okay, it is reduced, and also with glucocorticoids. But insulin, on the other hand, obviously opposite to glucagon, increases the activity of the enzyme, and thyroid hormones also increase the activity, so we have more cholesterol. Right? Then we have cholesterol itself inhibits HMG-CoA reductase. So when you have too much of the product, the enzyme gets inhibited. And also mevalonic acid is another inhibitor. There are 2 ways that you can act on the, like, for example, covalent modification of the cholesterol is carried out in 2 ways. You can have esterification or deesterification. So esterification is carried out by 2 main enzymes. You have LCAT and ACAT. So LCAT is is lecithin cholesterol acyl transferase, and it is in the blood, while ACAT is acyl cholesterol acyl transferase. So lecithin cholesterol and acyl cholesterol. Okay, and the deesterification is carried out by one enzyme, which is cholesterol esterase, which is in lysosomes. And the ACAT, by the way, is in the cells. A bit about the clinical aspects of cholesterol. You know, atherosclerosis is a disease in which plaques are formed in the walls of the major arteries, and the endothelial cells of the artery wall are injured, which is why there is a reaction. So calcification also occurs, and we know that atherosclerosis can lead to myocardial infarction, which is quite severe, and that depends on your diet or genetic. It can be genetic, like familial hypercholesterolemia, and diet, obviously fatty stuff. Moving on to the products. So products are bile acids mainly. So there are two types of bile acids. You have primary and secondary. So the primary ones are synthesized by the liver cells, and the secondary ones are the action of intestinal bacteria on the primary. Okay, moving on to the names. So you have taurocholic acid, primary bile acid, and glycocholic acid. You can also have tauro and glycoquinodeoxycholic acid, which will also be primary bile acids, but I think two are enough. And for secondary, you can have deoxycholic acid and lithocholic acid. They're both secondary, and obviously, as I mentioned, derived from the primary bile acids. So starting with cholesterol, of course, it goes to 7-alpha hydroxycholesterol, and the enzyme for this is 7-alpha hydroxylase, and this needs vitamin C, and also this is the key limiting or key step of the reaction. And from 7-alpha hydroxycholesterol you get the bile acids, first primary and then secondary, by the action of the intestinal bacteria, as I mentioned earlier. You can get conjugated bile salts from the acids if it reacts with either glycine or taurine. And the bile acids, they're stored in the gallbladder and released into the intestine. And if there is something known as enterohepatic circulation, and this is when some of the bile acids are reabsorbed in the intestinal lumen and brought back to the liver. So that's what it means. And the intestinal bacteria deconjugate the primary acids, removing the glycine and the taurine residues. So they are less soluble and therefore less readily reabsorbed from the intestinal lumen. Lithocholic acid is the least soluble bile salt. Moving on to the conversion of cholesterol to specialized products, we have the steroid hormones. Cholesterol is converted to progesterone in the first two steps of synthesis of all steroid hormones. And for this, it requires 4 cytochrome P450 enzymes, which are important. And it occurs in the inner mitochondrial membrane. We shall start with androgens, which have 19 carbons, and they're synthesized in the testicles, and an example would be testosterone. Then we have estrogens, which contain 18 carbons, obviously in the ovary, and one example is estradiol. And then we can have glucocorticoids, which are in the adrenal cortex and have 21 carbons. They have different locations. For example, cortisol is synthesized in the adrenal cortex, but in the zona fasciculata. And you need to know about their functions as well. You know that cortisol is involved in regulating the protein and the carbohydrate metabolism. The mineral corticoid aldosterone occurs in the zona glomerulosa of the adrenal cortex. Its function is to promote sodium resorption. Progesterone is synthesized in the corpus luteum of the ovaries. Estradiol controls the menstrual cycle. Testosterone is in the Leydig interstitial cells of the testes, and they're important for the development and function of the male gonads and determining the secondary sexual characteristics in men. So what's important is that cholesterol is converted to pregnenolone and then to progesterone, and the conversion to pregnenolone is the key step in this reaction. Moving on to the last part of this video, we'll speak about the conversion of cholesterol to calcitriol, which is a derivative of vitamin D. It is synthesized in the kidneys, but not throughout the synthesis, basically. So it starts with in the skin and then goes to the liver and then finally to the kidneys. Calcitriol promotes the resorption of calcium in the intestine, so it increases the calcium concentration in the blood. It is carried by transcalciferin, and so in the liver it goes from cholecalciferol to calcidiol. In the kidneys it goes from calcidiol to calcitriol, and before that in the skin, with the help of UV light, it goes from D7 dehydrocholesterol to cholecalciferol. So in the kidneys it is in the proximal convoluted tubules, and this is how you get calcitriol from cholesterol. And that would be it for this video. Take care, guys.